This is a Founding Media podcast, produced at Austin Community College District. Welcome to Science at the Mall, y'all. I'm your host, Dan Dillard. Even before COVID-19 took control of the world, people were still worried about viruses. Viruses are tricky and complex, and trying to get tested to see if you have one can be a difficult process. This is where Alan Blake and Dr. Richard Crockett come in. After a successful career pioneering biotechnology, the duo decided they needed to give back. Through their discussions, the pair wondered why there was not a rapid, inexpensive consumer device that had the ability to test for infectious diseases. This is when their company, NuClean, was born. While NuClean's original focus was on testing for Zika, it was realized that the product could be used for just about anyone to test for viruses. Early this year, it became clear that their focus should be shifted to COVID testing. So let's get to it and hear how Alan and Richard got where they are and what they're doing now. I'm excited to talk to you both, and I'm really wanting to learn more. This, as we were talking the other day, what amazed me is how long you guys have known each other. Would you talk to us about how you two met and what started your entrepreneurial journey together?
Sure. So Rich and I first met in 6th grade, so it's quite some time ago. Kind of went through the very similar experiences in terms of our education and small business type opportunities here and there, somewhere between age 10 and age 18, and it was just a great experience. We grew up in New York, so kind of northern Westchester area.
I think we connected, Dan. Really, we were both, I think, very early on entrepreneurs at heart, and that was something that was pretty obvious in the other one. So that was a natural bond. Even we probably wouldn't have used those words at the time, but certainly similar interests. So, well, why couldn't it be this way? And should we try that? Those kinds of questions. After high school, my interests were really bifurcated between computer science and biological sciences. Both were very interesting, and they had a lot of parallels in my mind. After my undergraduate that I did molecular, cellular, and developmental biology, did a little bit of research while I was doing that. That's when I came down to Austin, joined back up with Alan. We did the online endeavor that we talked about. And that was originally supposed to be electronic books. And everybody looked at us like we were nuts and said, "What are you talking about? What does that even mean?" And so not the right time for that. That was a lot of fun. It was an environment where there's a lot of energy. And I think when you're young and you don't know what you can't do, that's a great time to do that. It's a great time to take a shot at those things and to learn those things, because there's really no other way to get that experience other than just diving in and trying it. And so I love that. Learning,
and it sounds like, the dotcom was, for many people, unless you were like, one of the ones that actually succeeded, was just a learning experience. It was just one of those things where you dive in, learn, and fail. But fail is just part of the journey, right? So that's what it sounds like.
Well, ours was interesting. It was kind of in between. We were, I think, very successful to a point. We raised about $4 million. We got the company to roughly $12 million valuation, had customers at 75 universities across the country, had about 50 employees. And all of that, it was really the kind of first year, maybe closer to a year and a half, was trying to figure out the business model, probably hundreds of dollars or a few $1,000s in the bank, to this raising $4 million and a $12 million valuation in 75 universities across the country, to essentially back to zero, all in about 13 months and 50 employees, as I mentioned. So it was a really wild ride, and learned a tremendous amount from that. We ultimately the dot-com bust just came at a time where the people who had funded us just sort of said, "Okay, well, we've had enough of the dot-com days, and we're not going to continue to fund this." And our burn rate was just too much. We just didn't have the time or the resources. Really, over the course of literally 72 hours, we went from a bustling office with lots of energy and lots of excitement, like on a Friday afternoon in January 2001, to all of a sudden Monday morning, after some intense weekend conversations, okay, we're going to have to liquidate the business because there's no more money, and paid off all the vendors as best we were able to, as quickly as we could. and liquidated the business. And that's where the GloFish story started, which was the next 16 years. And that's the business that Rich and I started together. And it was quite an experience, which really directly led into what we're working on right now.
Tell us more about how you went from a dotcom to GloFish, and what was the thought processes there?
We didn't know anything about dotcoms when we started that either. But the original idea was, that we're still interested in doing something different and novel. And I had, through my exposure in doing different research, I've been exposed to different fluorescent proteins that were used in labeling studies. And so in science, you use these proteins, and some would glow red and others would be green. And you'd see an image and you'd see these really vibrant, beautiful colors. And the thought was, could you take those same proteins, and if you were able to express them the right way, not label individual cells, but label all of the cells in such a way that you could actually see the organism glow and fluoresce. Aquarium fish were really a natural fit because aquarium fish, the whole industry is driven on novelty and color. These things came together as an idea, and we started talking about it, and it certainly sounded like a very unusual idea. But after we were talking about it a little bit, it started to catch on in our minds and grow, and we decided to actually go and do it.
Wow. And so that's the beginning of GloFish, and as you said, 16 years later.
Yeah. It was really an extraordinary business. Essentially, the intersection of biotechnology, politics through all the regulations of biotechnology and genetically modified organisms and such, and aquarium fish. We ultimately were able to grow that to about 15% of the industry in terms of dollars spent.
Wow.
It was great. We had over 7,000 retail locations, so it included Walmart, PetSmart, Petco. We ended up selling the business to a longtime strategic partner in 2017, Spectrum Brands. We met a lot of other people who we got to work very closely with along the way, and learned a tremendous amount about commercializing biotechnology. And in the interim, one thing that is also really interesting, which sort of makes all this work and makes a little more sense, not long after we got going with GloFish, Rich had a chance to go to medical school and learn a few things about the medical space, which is obviously very important with what we're doing with NuClean today. So anyway, Rich, I'm maybe happy you should talk a little bit about your experience there, because obviously that relates to what we're doing with NuClean.
Yeah, so I actually went back to Yale to get my MD, did an extra year of research there, and that was a great, I think, insight into the difference between the theoretical world of medicine and the practical clinical world of medicine. There's a good quote from Yogi Berra that I like and I repeat not infrequently, which is, in theory, practice and theory are the same. In practice, they're different. And so there's a little of that going on when you're trying to deal with real patients and real disease states, which I think is very helpful, certainly, as we try to contextualize what we're doing here with NuClean.
So I'm going to go back to 2017. You had this exit. Everything was like great experience, did a lot of learning, good things. Now you're sitting in 2017, and how does this inspiration start for NuClean?
A lot of it was we learned so much during the GloFish business about commercializing biotechnology. What's really interesting is that in biotech, you have to really line up so many things successfully in order for it to work. You've got the technology, the science of it that needs to line up with all the intellectual property, which needs to line up with all of the regulatory requirements, which needs to line up with all of the commercialization requirements, including things like public relations and distribution, and then ultimately sales to the end customer. So this is just all set of things where if any of those don't work out, you could be in a lot of trouble in business. Right. And biotech is it's so cutting edge. It's really unique in terms of how some of these things need to be approached. And so, there we were. We learned all this. We sold this business, and it was a very successful sale. We were very fortunately just a lot of luck, a lot of hard work, and a lot of luck. That's, to me, success is really both of those. You need really a lot of hard work. I mean, many years of 60 and more like 80-hour work weeks for years and years and years, but a lot of luck along the way, because lots of people work like that and just for whatever reason, circumstances are different and can't quite make it happen. So you put all that together.
And great people. I think having people along with you on that journey that are also willing to make those sacrifices and willing to believe in what you're doing, it's so key to what those three ingredients, I think, together are the special sauce, if you will.
Absolutely. It takes the right team and being able to attract and really align everybody along the way. And so we just learned all this, financially did very well with it. So we're at a point where we didn't need to work anymore, but also being 40 years old, sort of having this feeling of, okay, well, I do want to do something meaningful with my life. I do want to do something, sort of get this whole next chapter of my life, and wanting to do something really helpful and kind of make a positive impact in some significant way. And, just feeling both the opportunity and the obligation, at least in my case, to say, like, look, you've got all of this that you've been able to do and potential and resources. Like, you got to do something with it to try to make use of it.
Is it more like a responsibility that it's like I feel responsible to now give back or do something different?
Definitely. At least, and everybody's different, but for me, certainly that was it. And so,
and we did, just to note it, in GloFish, we did a lot of work with clean water initiatives. Really, Alan spearheaded that idea, and it was fun, just phenomenally successful, where we were able to put in new wells and fix old wells in sub-Saharan Africa, and sort of lives saved per dollar that we were able to do there was unparalleled. And the organization that we work with, WellAware, who's also based here in Austin, at this point, still, I think to this day, has 100% success rate with the wells that they put in, and it just changes the whole environment down there. So that was all because of GloFish and the tie-in with fish and water, and our desire to do that same type of meaningful work for people.
That's great.
Yeah. This actually was really interesting. Just to mention it. So we gave a bunch of, it's just all the little connections. So our first marketing manager for GloFish ended up being sort of, I think, one of the first volunteers for this organization called WellAware. And so that's how I originally got connected with them. And over the years, they've been amazing. So they have got this just incredible track record. But here we are, very entrepreneurial. And I said, well, clean water we take for granted with the aquariums, right? We wouldn't have an aquarium industry if we didn't have clean water here in the United States. So people need clean water too, and so let's support the opportunity with WellAware to try and create some additional clean water infrastructure. Well, it was great. Rich got behind that. The board got behind that. And we talk about people and the ability to make positive change, even as a business, even if it's not the core business. And so then we took a look at this and the details, and Rich said, well, there's so many wells out there that people build, and then, you get a 5-cent piece or a 30-cent piece that breaks and the well gets abandoned because, in the middle of, Kenya or wherever it may be, you can't find a 30-cent piece. And so the well stops working. So, what if there were a way to just sort of go in and fix those wells, just do essentially renovate them and get them working again. And WellAware, to their credit, they looked into that, and the lady who runs it, Sarah Evans, is just amazing. And so they started working on that model where possible. And so the money we gave today supports something like 45,000 people in terms of clean water. Yeah, it's just a huge. And as Rich said, all of their wells are still working, and it's just been this enormous success. And that's really to WellAware's credit. But it also shows the opportunity with community and networks, and when people work together and look at things in a different way. And so, as Rich said, we had some exposure to this, and that was, at the end of it with GloFish, we both said, gee, with all the things we've done, this is one of the things we're most proud of. And so then the question was, okay, all that background, what do we do from here? And around that time, Zika was an issue in Austin, and my wife and I were thinking about having a baby, and she kept getting bitten by mosquitoes, and I was nervous about, well, are they carriers of Zika or not? And so then we were, sort of like trying to figure that out, and you'd have to go and see if this, what information did the city of Austin have, and not much. And anyway, it just really occurred to me that this was an unnecessarily difficult and frustrating situation, and why couldn't we just have a very rapid, inexpensive consumer device that would be able to tell you if the mosquitoes in your backyard were carriers of Zika? And Rich and I started talking about that, and pretty quickly understood that if you could do that, then you could really detect any nucleic acid sequence, any DNA or RNA sequence. And if you could do that, you'd have a infectious disease detection platform. And if you could do one that would be rapid and inexpensive and portable, wow, the implications of that are staggering.
Yeah, incredible.
Just staggering. And so in so many different ways, human infectious disease, animal infectious disease, genomics, to environmental DNA, to forensics, to just even research and development in general.
It always amazes me the initial idea starts with something that's affecting you or your family, right? It's like mosquitoes, kids, what do I do? And all of a sudden that turns into this much larger piece is what you guys are working on.
You sort of scratch your own itch, right?
Yeah.
Yeah.
Very cool.
Yeah. Well, and so we named the company NuClean, which was the name that was coined in I think 1869, Friedrich Miescher. Sort of the stuff inside cells, and that was I guess there's a bunch of stuff he was looking at, but part of it eventually was understood to be nucleic acids, so genomic DNA and of course RNA, and so that's where, as far as I know, where nucleic acid is originates from. And so
I was going to say, in a nutshell, a testing platform, and then tests that you can put into the testing platform to test for various things, correct?
Yeah.
The central idea was that it would be something you could use anywhere, that a layperson could use it, that it was going to be so easy, and those types of things. And so obviously how you apply that technology into different areas can be very differently situated. But the general idea was that there was such wonderful technology that had been locked inside labs for many decades, and it was all known. There's nothing overly complex that people didn't know how to do, but you never were able to get that out where it was actually needed. People talked about bedside tests or penside tests in the veterinary world, but they were always a stone's throw away, or they were formulated in a way that it just didn't really do what needed to be done. So we were really interested in bridging that gap. And the corollary to that, obviously, that was very meaningful, I think, to Alan and me as well, was the idea that if you could do that, then what that opens the door to is that technology can be deployed into the developing world. And so you have a situation where if you have basic ability to do diagnosis, you can make a huge difference in patient outcomes in the developing world. And to be able to have cutting edge molecular diagnostics that you could throw into a backpack, a whole bunch of these tests. You don't need electricity. You don't need refrigeration or cold chain. You don't need skilled people who are technicians to use this. And it does everything to the same type of standard that you'd see in a lab. That was just, the idea that you could do that was so appealing. The visual I always is this clock on the wall where, lives saved is wheeling around too fast for the numbers to see, that kind of thing. I think, no matter what else happens in my life, if I can walk in and that type of thing is a part of what I've contributed to, man, I know I'll put my head down on my pillow and feel like, that I've done what I came here to do.
Yeah. So something, as Alan's put it, meaningful, giving back to the world. Not that you hadn't done that before, but this is truly, especially with what we're talking about or what has affected us this year. We're doing a few episodes, as you guys are aware of, on COVID-19 and the impact it's had. It's just been something that's taken the world by storm. And so you went from Zika to starting working on shifting the focus to designing it for a test for COVID. So how did y'all decide to do that? At what point did you decide to do that? Yeah.
So just to be clear, the Zika was really what originally motivated the idea, but it was very quickly obvious once we figured out, okay, we're really talking about here as a platform, that there were better uses for that platform than Zika. And Zika, thankfully, Zika did not ultimately become a big concern in sort of the pandemic sense. Zika is not something you hear people generally speaking concerned about these days, or if people are concerned about it, it's obviously not nearly at the level of COVID as an example, or even the flu. How many people die every year from the flu, very tragically. So, and I don't know the current status of Zika, so it's I don't know, Rich. Do you know what is the current status of Zika? It's just seems like everything is so focused on COVID right now. A lot of other things are, which is one of the tragedies of COVID. If we think about it, it's not just COVID. I was reading an article not too long ago about how severely the COVID situation is going to damage work on so many other very important issues. Because as Rich said, if somebody's affected by this, it's really for each person, it's a tragedy, and it's if you're the one who is affected by something, then it's your entire world. So and that's why the platform is so important, because it allows us to work on so many different things. And ultimately, the goal is not to prioritize. The goal is to have a platform that can address all of these things. And that's really what is the promise of this type of technology.
As Alan mentioned, the transition was a transition in thought at the very beginning, where we realized that this was what we needed. I think, and we proceeded down that road for quite a while, several years actually, developing the platform, getting prototypes where they needed to be. And we were actually right on the cusp where we said, okay, now we really need to figure out what our go-to-market strategy is, and what is our first product, and what do the assays look like. And within about a few weeks of discussing that, it became very apparent what that product was going to be, because we were in January and February of this year, and we said, okay, well, now we know what we need to do. We're really very well positioned for this in terms of the device, and we think that we can really help people if we can execute well on the plan that we need to execute on.
I've heard so much about the different kinds of testing for COVID, right? And one of the biggest complaints that you hear is the nasal swab up the nose. Yeah. Would you talk about some of the challenges for the type of testing for COVID, and maybe some of the types of testing that are being done now, and how this improves it?
Yeah, there's a couple different types of challenges. One of the type of challenges is that you might have a very simple process, but if you want 100 million people to do it, it could be as simple as tying your shoes, and it is very hard to organize that. It is very hard to get manufacturing ramped up to supply that. It's hard to get people trained. It's hard to handle the logistics of it. And so one of the challenges for COVID is that it is such a large problem. It affects so many different people in so many different ways, and it has been new. And so our understanding has evolved over time. And a lot of the information that we used early on to evaluate how to manage COVID, both in terms of policy and in terms of medicine, was predicated on our experience with influenza outbreaks or other similar outbreaks in the past. And this has really been very, very different. So I think that's been a major challenge in terms of the testing. At the very beginning, it wasn't entirely clear where this was going to go. The models that were used to predict things are very sensitive to the inputs. And so, there's an old adage that all models are wrong and some are useful. And it was very difficult for people, I think, at the beginning to really fully understand what this was going to be. And even now, as we sit here today, a lot of things that we're going to say are probably wrong. All facts have a half-life, and there's no exception to anything that we know today about COVID. In terms of testing, knowing where the virus is in the body, in the nasal cavity, in the oral cavity, in the blood, and being able to validate tests that do a good job of not only identifying when the virus is there, but identifying when the virus is not there. And having those tests be available in a format where you could deploy that to many millions of people very rapidly, right, without experiencing bottlenecks in either manufacturing, supply chain, or technicians to operate devices. That's a very, very difficult challenge. And we're still struggling with it today, and we're going to continue to struggle with it for the foreseeable future. I think in terms of types of tests, broadly speaking, there are two types of tests that that we generally use. One is an antibody test or an antigen test. And what that does is it looks for the actual virus, but it doesn't amplify that virus, which is to say you need to have enough of the virus present for the test to be able to detect it. So when the test does detect it, it's very specific and it's a good test in the sense that you know that it is there, as long as it's been designed well. But sometimes it misses it. And that's why if you look at a urine pregnancy test, which uses the same type of technology, you have to wait sometimes 3, 7, 10 days until you actually get a result on there, even though you're pregnant the whole time. And the same thing happens with COVID. You can be infected with COVID. You can be shedding the virus, but you won't come up positive in some of these tests until you've gotten far enough into the disease course.
Rich, just to clarify one point, that specifically the idea that the virus has to be present, that would be antigen testing, whereas antibody testing is similar in terms of some of the accuracy considerations. Antibody testing is looking for your body's response to the virus, which also takes time to develop. So somebody could be infected and could be contagious before an antibody test would pick that up. And again, Rich's, I guess what I was saying before is his medical experience is really helpful in all of this because this is where you need to kind of bring together the hardware, the software, the biology with medicine. And then, of course, there's FDA and commercialization. And that's where all that other experience really comes in handy.
Makes sense. Thanks for clarifying that.
Sure. On that point, that's one general type of test. Those are rapid and those are inexpensive. Antibody antigen, they're generally clumped into the same general kind of category as rapid, inexpensive testing. They're also generally perceived as not being as accurate as molecular diagnostics. And molecular diagnostics would be the most common molecular diagnostic test is the PCR test. And other types include isothermal, and those molecular diagnostic tests are generally dramatically they're a lot more complicated. So those are the ones where you generally need to send a sample off to a lab, and it can wait a week or 2 weeks. It really depends on the available resources. And there's this really unfortunate dynamic where when there's an outbreak, what may have been a 1 or 2 day wait time, it can often become a 1 or 2 week wait time. So just when you need it the most, that can be more difficult. Now, to be very clear, there are cases where you can go to a hospital, for example, and get a test result a lot faster. There are some, especially now, that can be done more overnight. There are now kits that people can get sent to their homes, and they can provide a sample, and that kit is sent away. So there are different models for this, but they fundamentally all have the same basic challenge: it's expensive, it's time consuming in the process of from the sample to the result. And it generally requires people to go to a location, which if they don't have COVID and there's an outbreak going on, that's the last thing you want them to be doing is going to a location where people are getting tested for COVID. Or if they do have COVID, you also don't want them to be going to a location with a lot of other people. So there are pros and cons to these different types of testing methodologies. Ours, really, the idea of this was just to get the best of both worlds. So rapid, inexpensive, and have it be able to be used by a non-technical user and provide a result in under an hour. So the idea is spit, push button, get result, and that's a lot more. All things kind of considered,
sounds so much more
better
efficient
solution. Yeah, and of course, I should also clarify we're still in the development process here. So this is something that we're working very hard on, and makes a lot of sense if the technology can be brought to market.
Certainly, a lot better than a 6-inch swab, up
your nose,
tickling the center of your skull, basically. I don't know if you've had that, Dan. I've certainly had that and administered that, years ago. And nasopharyngeal swabs aren't much fun. And the good news is, in the sense of a diagnostic sense, that we can detect COVID-19 equally well in saliva, and that's been fairly well demonstrated at this point. The bad news about that is that's because the virus often produces a lot of virus, and that's one of the reasons that it's been spreading so effectively in the population.
So if this can be done with saliva, why isn't it being done?
So Dan, I think that's a great question. And one point to clarify is that it is being done with saliva with certain tests, and other tests use swabs that are more from the lower nostril. So the reason there's a lot of talk about these nasopharyngeal swabs is that's where the testing started, and that is probably still, I think, where the majority of the testing is. And I think it really is a question of the legacy testing and legacy development. So when COVID first started, there was a higher level of confidence that, or at least the belief that you needed the nasopharyngeal swab. So lots of companies went out and developed those tests. And once those tests were developed and those resources were expended and those tests were cleared by FDA, well, you can imagine there's not a huge amount of incentive for companies to take what precious resources we have around testing and now redo everything and go back through the FDA. And sometimes their platforms are just not amenable to it. The testing has to fit into existing platforms, and some of the platforms require nasopharyngeal swabs. So I don't know the overall percentage of what's nasopharyngeal versus what's saliva versus anterior nair, I think is the term for the lower nostril. I've heard about cheek swabs. I've heard about blood tests. I'm happy for Rich to add anything to that, but there is a whole variety of testing out there. It's just not that efficient for us as an overall society or for companies individually to go out and redo all of the testing once they've got something on the market. Do you want to add anything, Rich, to that to clarify?
So I think with respect to saliva and nasopharyngeal swabs, the real focus, as Alan said at the beginning of this, was really getting tests out as quickly as possible. And there were the fewest questions with nasopharyngeal swabs. And certainly, it's not the end of the world to get an NP swab. And if you're sick or think you may be sick, you should definitely get one. We don't want to scare people off with those, but they are uncomfortable and can be uncomfortable. And but that was not the primary concern as we were trying to get tests to market fast as possible. The goal really was to do what we knew worked. Now, as we've been many months into this, and we've had time to do the research, time to understand the disease course here and how the diagnostics work. Now we understand that there are other ways to sample for COVID-19 testing, including saliva, including anterior nairs or mid-turbinate swabs, cheek swabs, other things that are being done that are effective. And so now that those are available, the other considerations like the discomfort from an NP swab, the fact that a person who is going to get an NP swab has to wear personal protective equipment, that there's a risk of exposure and so forth. Those things become more important considerations. And as we try to get away from the sickest of the sick being tested to getting tests out where we can test lots of people and push the replication rate of this below 1 where it starts to get under control, that's really an important aspect of the testing, is getting something that people want to use, that can be deployed quickly, and that is very effective at determining whether someone is positive for COVID-19 or not.
It's also simpler from a supply chain perspective.
Yeah, for sure.
You don't have to deal with swabs, and swabs were a limitation early on. So, and I think that's where the sort of commercialization side that I talked about earlier comes into this, where you look at this and say, wow, it's a godsend that we have this testing available. And as Rich said, you got to get out what you can as quickly as you can. But then to look at that and say, okay, well, here we are. If we're developing our product for this application, for this target, this particular pandemic, then what's the best way to approach this that's going to help the most people in as efficient a way as possible? And that's why we decided to go with a saliva test in our case. One other point to mention is that these tests are made to process particular types of samples. So if you look at the test physically, they're physically adapted for the particular swab that goes in them. Or if it's a blood test, obviously it's going to be a particular process to process a blood test. And so the big challenge is, when we come to saliva, is these tests are not physically designed to accept saliva. So that would require the companies to redesign it and then go back through the regulatory process with that. and there's a lot of resources in that. There's a lot of risk in that. And it's not as simple as, well, gee, the company should just go spend money doing that. Well, should they be doing that, or should they work on a combination COVID flu test, which a lot of them are doing? So, if you're the company and you're saying, how can I help the most people as quickly as possible? And do you take your resources and say, well, let's take an existing test that works and redesign the physical test and the device and then go back through the regulatory process, which just takes a lot of time because it has to be done right? Or do you say, well, you know what, we could take those same resources and now make it a COVID flu test? And the reality is probably a lot less resources to make a COVID flu test than what you would need to go back and redesign your entire testing platform, plus all the risk that's associated with that. So
yeah, that makes so much sense. I think that, as consumers, we're just thinking medicine more efficient, but we don't think about there's a business behind the medicine and the resources required, the FDA, the approvals. You've got to make these, there's people that are making these type of decisions.
Well, and it's not just dollars and cents. And I think that's important for people to understand. It's risk. We've been working on our platform for 3 years now. This is not the kind of thing where, you just say, well, gee, we just want to make more money, so we're not going to do this. It's the kind of thing where you can say, well, we can spend the next 5 years trying to do this, and it's not even going to work. Or should we focus our resources on deploying the tests that we have? So even, I would say, take the business part out of it, the profit motive out of it, and you would still see the very common sort of sense practical decision to say, if we want to help as many people as possible, as quickly as possible, stick with the tests that you have and find ways to make more of that, make those faster, make the functionality of that better by adding additional targets, like adding flu to COVID and that kind of thing. And of course, there is the business reality on top of that, but I think it would be probably not fair to say that would be the driving decision in this. It really is an overall, how do you get the best outcome for everybody as quickly as possible?
So that brings me to my next question. We talked last week, and one of the things that just amazed me is so much misconception out there about COVID-19. And what are some of the things that you'd like to share, or could you share about these misconceptions and what we know or what you guys know as you're studying this and the testing versus what I consider political news?
Well, that's the first thing is unfortunately this has become an incredibly partisan issue, which is I think really unfortunate because there's a lot of agenda driven reporting and information and misinformation that goes along with that. So that's the first thing is just to recognize that that's happening. The other thing I guess I would say, and then we'd love to get Rich's thoughts on this, is that there's not going to be a silver bullet for this. There's this idea that, and I'm not sure to what extent this idea is commonly held. I think it maybe was more so not too long ago, but I'm starting to get the sense that maybe there's more of a realization of this at this point. But there's not a silver bullet. There's not all of a sudden we're going to wake up and there's going to be a cure, or there's going to be the vaccine solves the problem and we're done. You look at the history of vaccines, and it's really it's incredible technology and it's incredibly helpful, but it's a tool. It's one tool of many. The flu vaccine is great and it saves countless lives every year, but people still catch the flu who've been vaccinated. And part of the issue is not everybody gets the vaccine. Some people can't for health reasons. A lot of people choose not to. Of the people who do get the vaccine, it's not effective for everybody, certainly not equally effective. And so then you look at, okay, so people are still going to get infected with this. Well, then you have to look at therapeutics. Well, therapeutics are great. We're already seeing better and better therapeutics, and there's different kinds of therapeutics. There's antivirals and there's antibody treatments, and there's all different steroids and different things. But again, they're incremental. And so testing is part of that.
Real quick, since you touched on therapeutics, and I know this has been, one of the things that Trump said here lately about therapeutics. Can you explain to the audience what the difference between a therapeutic and an antivirus is? or vaccines?
I'm going to kick that over to Rich there to give the medical response for clarity.
For what it's worth. Yeah, so I think there are those sort of four categories of things that you do. There's diagnostics, therapeutics, preventative social medicine measures, and then vaccines. And a therapeutic or clinical management of a patient is really, it's a holistic approach. You look at the individual patient, and you are able to determine through your clinical acumen and the lab data you have and all the other things that flow into that, how do you manage this patient? And early on in the sort of disease cycle, no one had ever seen COVID-19 before. And so, again, you draw from other experience, and then the people that were responding to these things, they did a fantastic job with the information they had on hand, and they treated these patients in terms of how they were ventilated and how they were managed in ways that we now know are probably, there's probably better ways to do that. The idea that there's originally an idea that this was a cytokine storm like you'd seen in influenza, and now it may be, there's not to get too far in the weeds, but probably different systems, renin-angiotensin systems, bradykinin, all these different systems that are involved, ACE receptors, all these different things that we now start to understand a little bit better how these things fit together. And so what you do with the information on a patient, whether do you give them steroids, do you give them more oxygen, do you give them more pressure, what do you do with the tools that you have, that takes time to figure out. And always, you have a situation where you have small bits of data, and it can be easy to be misled to think, well, I did this and something happened, that must have been the cause. But it's really just a correlation and not a cause. And until you get enough road underneath you with a disease like this and get the data you need to make informed decisions, it's very difficult to know for sure.
Yeah, Dan, I think what the short answer to what Rich was saying is antivirals and antibodies are types of therapeutics. Therapeutic is therapy.
I didn't answer your
different
question.
Ways. Well, I don't know. Sometimes you ask a doctor a very straightforward question. You get anyway.
So I'll answer it straightforward. So an antiviral usually interferes with some process that the virus is doing, replicating, binding to the membrane, doing something where you're slowing it down so the body has a chance to kill it, or you're actually stopping the replication. An antibody, so they have monoclonal antibodies, convalescent serum, a whole bunch of different things, is actually the result of an immune process that has happened in response to the virus that you take from somewhere else. You take it from a goat or a rabbit or a patient that was sick, and you purify those antibodies, and then you are basically giving your body a head start on what it would normally do with its adaptive immune system, which is to produce those antibodies. So those antibodies are what your body produces, among other things, to try to contain and stop that virus. And so you're basically equipping your body with a tool that is still usually a week or two away for your body to be able to manufacture itself, to give your body enough time to catch up, understand this virus itself, and mount an effective immune response.
So I've learned so much about this from you guys. So thanks for sharing that. I want to ask one last question, which is, we're doing this on Science in the Mall, y'all. Tell me how that's benefited working with them and starting this new company.
Yeah, it's been really wonderful. And ACC has done just a great job of creating a bioscience incubator where places people can start. I think there's a lot of great science that goes on in labs where transitioning that out into the real world is very difficult. There's some industries where it's not difficult. If you want to create an app, for your phone, you can do that by yourself with a laptop. Right. But if you want to do biomedical sciences, you need a lot of equipment. You need environments that are suited to that, and there's a substantial cost to starting up that endeavor. And that's okay if you're really confident about what you're doing, but there's so much that is uncertain at the beginning of these things that having the ability to go into a place like ACC, where they've got a whole bunch of different equipment that you can use, the facilities, is really great. Because as an entrepreneur, what you want to do is you want to go in there and fail really fast and figure out that you should go do something else if that's going to be the case. And so,
what you said reminds me of just de-risking. Every time a VC or anybody looks at a new company, it's like, okay, how do we de-risk this as much as possible? And it sounds like what you're talking about is this type of business is not something you can do in your garage. You got to have all this equipment, and one of the risks is putting that much capital down. And if you don't have to, then you're de-risking.
Extraordinarily expensive. And Rich said it's okay if you have a lot of confidence. The other component of that is, and your investors have a lot of confidence. And the reality is, the reality is it's just not going to happen that way. Right. So it's more like if you and your investors have a crystal ball. And so ACC is a tremendous resource for the community. I really couldn't overstate that. It's a tremendous resource because it allows people to get into a lab and start doing real world-class science with world-class equipment and the community that's there for, it's pennies on the dollar to what somebody would otherwise have to spend. And, Rich and I used the word fortunate before with our GloFish business that it worked so well. We were fortunate coming into this because we had the resources and we had the network to really give ourselves a dramatically better starting point than what we had with GloFish. But even that, without ACC, it would have been pretty daunting to look at the investment. We recently moved into our own lab, and it is very expensive, not just to set up the lab, but to maintain the lab. And so having that as a shared community resource is tremendous. It's sort of like, the difference between paying your monthly electric bill and having to go out and start the electric company if you want to have your light bulbs turned on in your house.
Makes sense.
Yeah. So anyway, really appreciate what they've done, and the leadership there is phenomenal. Nancy Lyons is obviously running it right now, and she's just been incredibly supportive, and that's a big part of it too. I really want to stress that it's not just the infrastructure; it's the people and the leadership and having their support. That when science is cutting edge, and you sort of have constantly have to adapt and adjust, and having leadership there who can understand and really support that is very important. And I think it made a material difference for us for sure.
And who's aligned, who's really behind you, wants success. It feels like many times people have their own agendas in different aspects of life. And getting them to be aligned to what you're doing, to pull in the same direction, just not to be in your way is an effort, but to have someone who's a partner, and it says, what can we do to help, and happy to make connections and really acquiescing to any reasonable request is just, it's refreshing and wonderful. And I think there are a lot of companies coming through there that ordinarily would stay ideas, not get a chance to prove that out before they went out to write the bigger checks to build, labs or what other facilities they need.
Which obviously would the failure rate would be much higher because they wouldn't get the money, right? So
the best companies don't always get the funding. Best ideas don't always get the funding. Sometimes these things get locked away, and it takes a lot to make something happen. You have to get everything right to make it work. Even one thing's out of place, usually that's not a good outcome. So ACC is one of those things that, by being there, really lowers the risk and the bar to actually trying out some of these things and vetting them out before writing those bigger checks.
Makes so much sense. Alan, you reminded me of something you said a second ago. I was interviewing a scientist from Austria, and he said, the correlation between entrepreneurism and science, the thing is, I don't like the word failure. Entrepreneurs use that word all the time. We just call it testing in science because you just have to test. But tying it all together is you have to fail often, fail fast in these tests to know what works and what doesn't. And for that, you need the equipment and so on and so forth. So, guys, I really, really have enjoyed talking to you both today. I think it was such an informative podcast, and I'm sure the audience will benefit from it as well. Thank you so much for your time.
Thank you. Great to be here. Thank you, Dan.
Dan, it was a pleasure.
Thank you again, Alan and Richard, for joining us today and teaching us more about the world of biotechnology and viruses. The work you are doing is fascinating, and I'm wishing you success in the future of NuClean. If you'd like to learn more about NuClean, please visit the link in our show notes. Science in the Mall, y'all, is created in partnership between Founding Media and the Austin Community College Bioscience Incubator. To learn more about the ACC Bioscience Incubator, please visit the link in our show notes. If you like what you hear on this show, be sure to subscribe and share it with a friend or family member.